This chapter is going to go through modern applications. Now, I'm just going to talk about some of the stuff in here, but I highly recommend watching the videos that are linked below, just because this technology changes frequently. So I will test you on certain things, and I'll make sure you know what those are as we're going through this. So really, the big piece of equipment that is huge for making these sections of DNA that are used in recombinant protein synthesis is a thermal cycler. So this is going to be something that is used for PCR. Highly recommend watching the videos on PCR. So it takes a sequence of DNA and can make millions in just a few hours. And then that is what we call amplified, and then it can be used to detect diseases or used to make proteins for different things. So recombinant technology is the artificial recombination of DNA from two organisms. So like for human genes like insulin, people that are diabetic aren't making it. And so what they did was they took the gene from a human cell that codes for the insulin protein, and they cut it and they put it into a plasmid, and then they put it into E. coli. And then E. coli doesn't know what it is, so it's just going to make that protein over and over and over again because it just does that. So then it is able to secrete insulin, and then we can gather it up and then use it to give to people. So any gene can be put in there depending on the size. Sometimes you've got to use yeast because they can handle some of the larger genes, but if they're small genes, then bacteria can use them. Sometimes you have to use a eukaryote because they do that post translational modifications that need to be done that we talked about before. So one ways that they cut it is through the use of restriction enzymes. So this first one in A is one that I've used a lot, which is BAMH1. So BAMH1 will recognize what we call a palindromic sequence. So it notices this specific sequence, and then it's going to cut it on both sides. So it makes a sticky end. So you do the same thing. You cut the DNA with the same enzymes, and then you can glue them back together. Hey3 is one that makes a blunt end. I've never liked using the blunt ends because they could be put in backwards. So this one, you can make it so it goes in the right direction. So it's going to be read the right way. Puck19 is a plasmid, and it has this polylinker site. And so it uses a LAC-Z gene that will put a color change potentially. It's got Hindi3, PST1, Sol1, XBAL1, BAMH1, small 1, and Echo R1. I've used all of these. So you choose which one of these enzymes you want to use on both the polylinker site and the gene that you want to put in there. And it also has, so this LAC-Z reporter gene is going to be to where it will disrupt it, and it's going to change colors. It'll be either blue or white. And then it also has an ampicillin resistance on it, so that you know that the bacteria has the gene because it has a resistance to ampicillin. So you grow it in the presence of that antibiotic, and it will select for it. So this is what we call a vector plasmid that we use. So steps involved in cloning: you're going to take the foreign DNA and cut it with and the plasmid, cut it with like BAMH1 or one of your sticky ends. I like sticky ends because they are able to adhere in the right direction. And then you put it into the bacteria through transformation. You can electroporate it, which means you shock it, or you can heat shock it, and it's going to go in there. For this particular one, there's 3 outcomes. The bacteria does not take up the plasmid, so it's not resistant to ampicillin. This one, it takes up the plasmid, but it doesn't take up the recombinant plasmid. So it has an intact LAC-Z gene, and those are going to be blue. And then if it does take up the plasmid, it's going to be both ampicillin resistant, and it's going to be white because it cannot produce the beta galactosidase, which is blue. So we call this blue-white screening. So that's what I was talking about before. It's either going to be blue or white. Blue is bad, white is good. So we'd pick the white colonies that grow on the ampicillin plates. You can also make genomic libraries in much the same way, where you take the fragments, you put them in plasmids, and then you have all these different outcomes, and then you pick them up and you can sequence them and find the genes of interest. You can also use a recombinant phage. We talked about this in the last lecture, but you take the genome and you digest it, and then you just build your recombinant phage, and then they're going to infect E. coli. And then each phage is going to have a unique fragment from the genome. So we call this a phage library. Complementary DNA is from the mRNA of the retroviral reverse transcriptase. So it converts it into double-stranded copies, and it is inserted into either the plasmid or a bacteriophage, and you can produce the cDNA library, which means these are all coding genes. Electroporation, so like I was talking about before, you've got this plasma membrane, and if you shock it with an electric pulse, it opens up the membrane and allows those genes to get through. You can also heat shock it, and then they reseal itself. Remember that fluidity of the membrane is that way. You can also micro-inject, so you can use a needle and inject into eukaryotic cells. You can't do this with bacterial cells just because they're too small, but you inject it with whatever DNA you want it to have. There's also a gene gun. So what happens here, you've got a pulse of helium and a cartridge, and you've got the gun barrel, and this is to get past the cell walls in plants. So remember, those cell walls are pretty tough. So these gene guns will pulse through and get through that cell wall. And then that's what they look like in real life, gene gun. A TI plasmid, this is in Agrobacteria tumefaciens. It's a shuttle vector, and that's used in plant cells. So this one can actually do plant genes. It will be cloned into this TI plasmid and then introduced into plant cells. So the gene of interest then recombines with the plant cells and is able to grow transgenic plants. DNA probes can be used, so you isolate DNA from body fluid, denature the DNA by warming it up, and then combine it with DNA probes that are complementary to the gene of interest, and then it is going to be like a molecular beacon, and then the DNA is going to hybridize or bind, and then it's going to be glowing in some way, depending on what kind of probe you have. Gel electrophoresis is another one. Please watch the video on this one. This is very important. But you pour an agarose gel and a buffer in a plastic tray with a comb. We're going to do some of this in labs later on in the semester. The comb is put in there so you can get your sample in there. DNA samples are colored with a tracking dye so you can see where they're at. And then it is given a electrical current. So DNA is negatively charged, so it is going to go away from the negative and towards the positive. So you're going to separate it based on base pairs. So the smaller the base pairs, the faster it's going to go through the agarose. Imagine it being like a labyrinth. They've got to run through a maze. Okay, so the bigger, the slower they're going to be, and the smaller, the faster they're going to be. So then you've got a ladder that's got known base pairs, and you can see which gene you want because you know what size it is. Okay, so they use this for DNA fingerprinting, for finding, crime scene samples. They use ethidium bromide. We talked about this last chapter with the way that the ethidium bromide is going to intercalate into the DNA and bind. And then you put it under UV light and it's going to glow, just like this picture in C. I've done this a million times. But be careful, ethidium bromide is carcinogenic. RFLP sequences. So this is when you digest the chromosomes up, and then it's going to give you a fingerprint. So everybody's DNA fingerprint is going to be different. And so it's going to digest them and separate it, and you can tell which ones are mutated. So this is the normal, this is the mutated, or it can also be used for finding crime scenes, which is a lot of what happens. A southern blot is when DNA is first put through an agarose gel, so you separate it, and then you put it on a membrane, and then you can look at it through a DNA probe to make sure you've got the right one. So instead of having all this background DNA, you can find out exactly which pieces you want. I've done something similar to this called a Western blot with proteins. This one is gene expression or a microarray. Definitely cutting edge technology here. So this is going to show you what genes are expressed, either up regulated or down regulated. So you will look at different types of cells, and then you give them different probes, and you can actually see which genes are turned on and off in different scenarios. So this is showing cancer cells. So the green are only expressed in healthy cells, red are in cancerous cells, and yellow are in both cancerous and healthy cells. So you can kind of investigate what's happening on the genetic level in the cells when it's in different scenarios. I've done this when we looked at bovine macrophages and their exposure to different bacteria. They will up and down regulate genes when they're exposed to bacteria. It's pretty cool. SDS page, I love these. These are fun. This is how you can separate a protein, different protein profiles. So SDS is sodium dodecyl sulfate, and it is going to separate it and denature the protein so it doesn't have any charge. So it's going to straighten them out. And then you put it through an agarose gel, and remember, the bigger pieces are going to go slower and the smaller pieces are going to go faster. And you use this Kumasi stain, which smells amazing. It smells very I can literally smell this picture right now because I've done these so much. But you'll see a banding pattern, and then you can see which proteins are there. So this is for proteins. This bullseye rash. So this is from Borrelia burgdorferi. And so it's showing you this because a lot of these proteins are expressed during infection only. And that's what we did. We actually looked at protein profiles during infectious states. PCR is where you make a bunch of copies of a specific sequence of DNA. It goes through several stages. So in the first cycle, you denature it with a 95 degrees denaturation cycle, and then you do an annealing of a primer at 50 degrees, and then extension with a polymerase that is at 72 degrees is super hot. And so that means that you have to have a polymerase that won't denature in hot. So they use what they call a Taq polymerase. So it's from a thermal bacteria that can withstand these heavy or these high at temperatures. So from one round, you start with one and you get two. Cycle 2, you start with 2 and then you have 4. And then 3, you're going to keep getting multiple. You can do hundreds of cycles if you want. You can also have some DNTP, so you can use these to sorry, I've lost my train of thought. I got all excited about PCR. These are used to go into the DNA strand as a marker, and so you can actually stop DNA synthesis here. So these can be like an imposter kind of. You label them, so you've got different labels on them, and then you can look at this graph, and it'll show you where those are incorporated. Okay, this is Sanger's model here. This is showing sequencing data. Again, way far out there. Not going to spend a lot of time on this one. This one's showing PCR products of Clostridioides difficile. And so you've got different samples. And so this is like showing what a particular person, the type of strain that was taken out of this person. And then these are known strains. And you're going to match up the profile to figure out which one infected Javier. So it has to have the same exact profile. So 78, 126 definitely isn't it. 12 has a couple bands that are similar, 18 has a couple, 17 has a couple, but 27 has all of them. So your reference strain, the one that probably infected Javier, is probably 27. Okay. So again, you can look at it that way. This one is showing the different strains in a DNA ladder, looking at which pattern matches the one that is the culprit. Again, just looking at it in a different way. You can also look for green fluorescent protein. These are reporter genes. And so you can put these into different organisms to show expression. You can get genetically modified organisms this way, like glow in the dark cats and mice. You can also do blue-white screening. So this is a picture of the actual blue-white screening in real life. That we're going to just skip. This is silencing RNAs and microRNAs that will look at silencing genes involved in pathogenesis, looking at different sequences. Gene therapy, this one can kind of freak people out. So gene therapy is when you use an adenovirus to treat or cure certain genetic diseases with a patient with a defective gene. So you put the correct gene into a virus and allow it to infect. There has been some issues with this with cancers because they can't regulate all of the effects of the virus. So those are very closely monitored. Okay. So that's just a short list. Make sure you're looking at the videos that I provided in the announcement.